a big open question will be one of the goals of this naturalistic behavior field moving forward is how continuous is behavior, both behavior itself as well as its neural representation. The general idea is that motor cortex is saying what to do, and striatum is saying how to do it. And all of this is making it sound like I have thought out my future dreams of being
I had no idea we were going to talk about this. So.
Me either.
This is Brain Inspired. I'm Paul. And today, my guest is actually my current employer. As some of you know, I recently got back into the research world, and in particular, I work in Eric Yttris' lab at Carnegie Mellon University. Eric's lab studies the relationship between various kinds of behaviors and the neural activity in a few brain areas known to be involved in enacting and shaping those behaviors. Namely, those brain areas are the motor cortex and basal ganglia. And to study that, he uses tools like optogentics and neuronal recordings and stimulations in mice while they perform certain tasks, or in my case, while they freely behave, wandering around an enclosed space. So in this episode, we talk about how Eric got to where he is now, how and why the motor cortex and basal ganglia are still mysteries, despite lots of theories and experimental work. We talk about Eric's work on trying to solve those mysteries using both trained tasks and more naturalistic behavior. We discuss a little bit the challenge of even answering the question, what is a behavior? Which you would think would be a simple question, but it's not. And we discuss lots more. You can find relevant links in the show notes at braininspired.co/podcast/185, where you can also learn how to support this podcast on Patreon if you're feeling generous. You may notice if you're watching, I am not in my usual studio location. And that's because I'm at a conference, and now I must go mingle at the conference opening reception. So wish me mingling luck. I wish you luck in your studies and your endeavors. And I'm glad you're here to mentally mingle with Eric. Like me, you have a non-human primate neurophysiology cognition background. Very similar, actually, reaching. I didn't never do reaching, but I was all saccades, eye movements. And, you published some in eye movements. And unlike me, you've stayed in academia essentially. And, I went away and now I'm coming,
You're back into the fold.
Groveling back. Yeah. And you were, you took a chance on having me in your lab, but, I've never asked you how, your own personal take on your trajectory from those non-human primate world to, and you did some rat work and now mouse work. I mean, was this like a conscious decision, or is that just kind of how it flowed, or what?
Well, so the rat work was in my undergrad, but
it was. Oh, okay. Yeah.
Yeah. But so I had a great education working with primates, and Larry Snyder was the world's best advisor. But I thought a lot of the questions were getting sort of rehashed over and over again, and particularly, for the right questions, doing monkey work is absolutely vital. And in some ways, for some questions, you can only do it with non-human primates. And then there was the entire toolbox, including optogenetics, that was opening up for mice. And so for my postdoc, I interviewed at a couple of mouse labs, a couple of monkey labs.
Oh, you did? So you had that door open still?
I had that door open, and was just looking for good questions. And so what I ended up doing when I moved to Genelia Research Campus, they were in the process of doing some work training mice how to do a reaching task with a little joystick, a PlayStation 3 controller, with a coat hanger shoved in the end. And I thought, oh, great. I get to continue doing the reaching and what I think the non-human primate stuff, both the education and the sort of approach looks really in depth at behavior at a level that I think for rodents is coming online. But for years it was just lever presses.
Yeah.
And for reaching or saccades, you look at movement time, you look at trajectory and the variance of those trajectories and hypometria and all these things, these important kinematics that now are becoming much more popular in the field. But when I started my postdoc 12 years ago, were fairly uncommon for rodents. But yeah, it was initially I was trying to train mice to do a center out reaching task with 4 different directions. It did not work. But it's good to be ambitious sometimes. But so the idea was kind of keep the reaching thing going and ask some more detailed questions thanks to the trade-offs, the benefits of the model.
Yeah. One of the things that I want to ask from that is, so non-human primates, the reason that we study them is because they're as close as we can get to our brains and our cognitive abilities, while still being ethically okay, in this day and age. And so all, these tasks are being adapted for things like rodents, mice, but they have different brains and they have different cognitive abilities. It's not like a direct translation. And I know that there's a lot of excitement in studying. So on the one hand, there's a lot of people saying, well, mice, they're a lot smarter than we first thought or than we gave them credit for. And we can study these like cognitive functions in mice, right? And on the other hand, there's pushback saying, yeah, but you're not necessarily studying the same cognitive functions. They have different brains. The brains aren't perfect homologs from from an area, let's say motor cortex from a mouse to motor cortex of a non-human primate. Where do you stand in that? And have you sort of changed your mind at all through, your studies or?
So I actually, it's interesting you went that way with the question, because my first response was, you said, oh, we study monkeys because they're really close to humans. I've seen plenty of great human work on the area that I was studying in parietal cortex, and they have 2 area LIPs and 5 different divisions of parietal reach region. And monkey brains are as similar to human brains, roughly, as mice are, in that there's some general principles. Parietal cortex is one thing, occipital cortex is going to be doing another, but there's a whole lot of differences. And so I would have said, if you hadn't gone the direction you went, I would have actually said, we studied, or at least I studied, monkey behavior and monkey neurophysiology because of what a monkey can do, both cognitively and in terms of its behaviors. If you want to study in-depth attention, or certainly saccades or skilled reaching, you either need a task that is made accessible to a rodent, or for that matter, a monkey, or you just can't do it. And so for any question, you pick the appropriate model system. And so I'm not trying, when I was studying monkeys, I wasn't trying to understand the human brain. There are some ideas, for instance, parietal hemi-neglect. I was injecting muscimol and inactivating parietal cortex, and some of the things we were looking at were related to that, the parietal hemi-neglect in humans. But I was interested in monkeys. And now that I'm studying rodents, I hope that I'm uncovering some fundamental principles that apply to at least mammalian brains, if not vertebrate brains, very broadly glossing over the fine points. But when I study a mouse, I'm studying a mouse, and not just that. I'm keenly aware as we've moved into more and more naturalistic behaviors. I'm studying a super inbred mouse that typically is raised in a prison population.
Yeah.
And even within the inbred lines, there's plenty of nuances there. So I try and keep, I guess the word's not humble, but I know about the mouse that I'm studying and the neurons that I'm studying, and I try and keep it limited to that. If others can take those ideas and run with them, or we look at other people's data and applies more broadly, fantastic.
But is that why you got into neuroscience? So I got into neuroscience because you've been talking about brains and comparative, across species, right? But then there's the cognitive functions comparative aspect across species. And I got into neuroscience because I wanted to understand consciousness, et cetera, highfalutin stuff, right? And understand, higher cognition. And then one way to see my trajectory is just getting like lower level questions. And one of the things that you said, and we'll talk about naturalistic behavior, is there's still questions like we don't really even know what motor cortex does, not that it has like one function, right? But you get in and you think, well, I can study brains to study, in your case, like attention, intention, in parietal areas, for example. And these are cognitive things that you can't necessarily study in a mouse yet. Maybe they're a great model for attention, but you're talking about brains. Do you think the same way about cognition, or have your interests changed? And do you think that mice are a good animal model for that?
So I guess to come at it from my origins, even as a high schooler, I was lucky enough to know that I wanted the job I have today.
What the hell?
Way back when. Yeah, I missed out on the whole switching your major 3 times. So yeah, maybe even as early as late middle school, I loved music, philosophy and psychology, and biology. And so combined philosophy, psychology, and biology into a field that used to be called psychobiology, now neuroscience. And so it's always been about the brain and particularly behavior for me. If we can help treat diseases in humans, fantastic. Definitely a plus, and I'd gladly, and we do go into some models of OCD and Parkinson's in order to benefit mankind, and not just with knowledge, the little bits that we do. But yeah, I've always been at it for the brain. And with that, I don't even know, cognition carries a lot of weight to it. And I make the comment that I'm super jealous of people studying sensory systems, particularly, primary sensory cortices or on out towards the periphery, because they have such great control. In the motor system, basal ganglia, motor cortex, we're closer to the output. The people who study all the wonderful mess in between, my hat's off to them, because that is them.
You envy V1 people, but
yes, I envy V1 people incredibly for all the control that they get access to, but for the stuff between sensory and motor, the cognition side, that's hard to do. And some people do it fantastically, but there's a real lack of roadmap there, looking in from the inside. I kind of feel foolish studying cortical striatal circuits in a mammal when we can't even explain all the ins and outs of a nematode nervous system or a fly. And
so why don't you do that? Why don't you go to nematodes?
I only recently got associate. Give me a little bit of time. I do, if I had to jump, and actually the first experiment in the lab was I got some slime molds and was just going to do some computational modeling on their performance. But yeah, if I jump ship from mice, I would go slime mold or octopus, just because
that's so different. Those are two completely different paths.
They are both organisms that are capable of, I mean, maybe this is coming back at cognition. They have a surprisingly complex and adept cognitive ability, and comparatively far fewer moving parts. Slime molds are just a single cell, and octopuses are it's somewhat of a distributed circuit, but not nearly the neuron count of, say, even a mouse brain. But yeah, if I had to do that, I think I'd get wild and go with one or both of those.
Is that something that you think you could split your like? Could you expand your lab and have a half slime mold, half mouse, or third and third and third? Like, could you do that? Would you
certainly not third?
Okay, too much.
I mean, in the first 6 months while we're waiting to get things going, I did have the thought of, growing slime mold in petri dishes in my desk, and
you had them, right?
I had them.
So what happened? What you haven't told me this.
I take care of mice and my daughter much better than I happen to take care of slime molds. Okay.
Enough said. Gotcha.
Yeah.
Maybe your future is an octopi then.
We'll see. But I mean, in terms of some of these questions, we have two-arm bandit and probabilistic decision-making tasks going on in the lab. It might be fun to see if the models that we and our collaborators build, if we can get the same sort of things out of slime molds. And there's some good evidence that to a large extent you can. And so it's I don't see that being 50-50, but if an enterprising, probably more master's or undergraduate student said, hey, I want to play around with some slime molds and ask some cool questions, I would probably giggle with delight.
Well, see, I think you're saying
setting myself up. Yeah.
Undergrad or master's. Well, I think you're undershooting the dilemmas that will arise, or right? That because everything will be harder than you think it is, and it'll snowball and turn into a bigger project, I would imagine.
And I think that may be part of it. Master's students are in the lab no more than 2 years, and the typical undergrad, at least once they get going, is probably 2 to 3 years max. So if it's a terrible idea, there's a much shorter clock.
Okay. Fail fast, right? And break things. Yeah.
You don't know you're trying unless you break things every now and then.
All right. Well, so we haven't talked any real science. Oh, you know what? Let me just interject, because since we're on the topic of like animal models, and maybe we'll just kind of get this out of the way. I mean, one of the things that I've been interested in the past few years is, this recent push to use artificial neural networks to study brain dynamics and parts and functions and stuff. And it struck me in this conversation, this would be a relevant time to ask you your thoughts on, instead of using animals, using networks, right? And what we can glean from artificial networks. And I'm going to guess that you're not terribly interested in that approach by itself anyway.
So I tend to think of computational models, AI, deep learning methods as best used as part of a circle, a cycle. We ask questions of the biological brain. We find things out, and then either as a screen or to do a screen because it would be hard to do all these theoretical experiments in the biological brain, or just difficult generally because of some feature of the question itself. Try things out in an in silico model, whatever the case may be, and then use those results to make predictions to come back and ask a more intelligent and pointed question. And several groups are taking this approach. Again, there's lots of important questions out there. As someone who has always been interested in the brain, I'm going to be focused on the brain. And I know there's more, there are better solutions now to this issue, but I know a common complaint of artificial neural networks is you're replacing one complex system that you don't understand with another complex system you don't understand. That is not wholly true anymore. And I think there's some really cool things we can do with that, but I would still think that we would be, at least in terms of the questions that I'm most excited about, we're best served if we ask the hard questions, technically hard questions of the in silico brain, and go from that neural network and then use those results to ask better questions of the biological neural network. If we can do that.
It also, not to harp on the octopus and the slime mold, but so I think in terms of mammalian brains, right, and modeling mammalian brains, especially with like deep learning models, and I guess what I'm trying to ask is like, what question are you interested in? And maybe they're orthogonal, right? Because you said you're interested in the brain, but then which brain, right? Because an octopus brain is very different than a mammalian brain, and then there's a slime mold and stuff. So, and it wouldn't necessarily make sense to model. I mean, I guess you'd have to. How important would it be to set up? Okay, so let me back up. Like one of the successes of like deep learning models is that the architecture doesn't matter as much, right? You just give it a task and then it learns, and then lo and behold, it has some like dynamics that are similar to dynamics in whatever brain area you're studying and whatever cognitive task you're studying.
And hopefully universality holds up and everything works out great. Yeah.
But then if you probed an octopus brain in the same task, I would imagine you would get a different answer. I mean, I don't know. I guess you'd have to do it.
Maybe. And sort of circling back to asking the right question in the right way. I mean, my goal should probably be define more to brain and behavior, like things doing things. And that's why I've said despite all the benefits away from the sensory side. So, how does an octopus figure out which arm to reach out for a tasty crab, and how does it get the arm out there? How does it grasp it? How does it know where it is in space? And figure out which of the arms to use. How does it decide how to open up a jar, even though it's never seen a jar before? And so much of the allure there would be the particular differences. If I just wanted to understand how do I get an effector from point A to point B, unless I was a soft body roboticist, there was no way I would approach an octopus. And all of this is making it sound like I have thought out my future dreams of being
I had no idea we are going to talk about this. So.
Me either. Making it seem like I have plotted this out much better than just a few random conversations with people. I'm sorry we're going down this No. This is kind of cool. I hope somebody reaches out. And I actually chaired a session at SFN this past year, and there was a really cool octopus talk there. Maybe it's in the cards. We'll see.
All right, let's get back to what you spend most of your time thinking about. What you mentioned, that something was not the question that you're most interested in, or the questions that you're most interested in. Do you have a question that you're most interested in? I mean, that's an unfair question I know.
I think the overall extremely high level question is trying to understand how circuits produce the appropriate behavior. How do we transform thought into action? And so this encompasses decision making and sometimes, value estimation, motor control. So that's for the very broad question, I will give the very broad answer, but
Fair.
I think it's still appropriate in terms of what gets me excited, and that all these things, one way or another, fall underneath it.
So circuits, you study motor cortex and basal ganglia these days mostly.
Yep.
And of course, there's a motor cortex, basal ganglia loops. There's corticocerebellum loops and stuff. So these things are connected in loops. So it is a circuit, but of course there's a lot of divergence and convergence within these circuits. And of course, cerebellum itself is still a mystery, but
it's half the neurons in your whole brain.
It's more than half. It's
ridiculous.
Like 70%, isn't it? I thought
depends on the species.
What about the octopus, Eric?
Yeah. Pretty sure it doesn't have a cerebellum, but shows what I know.
Yeah, that's right. And I always think of that whenever there's an estimate for the number of total neurons. Like, we have so many neurons. I think, and they're all in the cerebellum, which is an interesting
basket cells, right? Or
Purkinje?
No, it's the tiny ones that send up the parallel fiber.
The stellates. No.
Someone's gonna
neuroscientists. We need to know this.
Oh, we got to edit this out because someone's gonna yell at us.
Yeah. Lots of neurons.
Lots of neurons in the cerebellum.
Well, that just goes to show you how neglected the cerebellum is too, right?
Oh yeah.
Although plenty of people study it, and a lot of people are saying these days, I'm hearing, especially from philosophers, for some reason, they think that the cerebellum is coming back into popularity. I don't know.
Oh, I mean, David Marr is thought to be a philosopher. He was a neuroscientist. I mean, philosophers or whoever will claim him. He's a computational scientist. Mm-hmm. I mean, thinking of Marr's 3 levels of analysis and all that, that comes out of largely cerebellum, at least as far as I understand.
Well, he had one of the early models for the cerebellum as well. Which was wrapped up in his Marr's levels analyses. But I mentioned the cerebellum because it's still a mystery, but so are, if you open a neuroscience textbook, it'll tell you motor cortex commands motor behaviors, and basal ganglia, depending on which textbook you open, either is a gate, which allows things to happen, or, it inhibits certain actions, and on and on. But we really don't know. I mean, we know so much, and there's both of these brain areas, motor cortex and basal ganglia, are still mysteries. I know that you have your own, would you call them hypotheses about, frameworks, hypotheses about what they do? Why don't we start there? Why don't you share your ideas about what motor cortex and basal ganglia are doing? And then if you can, just kind of pit it against a couple other ideas that are have been studied.
Sure. So I think the foundation of most, if not all, models of basal ganglia function and cortical striatal dynamics, including the cortical basal ganglia thalamic loop, are largely based on the work of Roger Albin, DeLong, and Crutcher and others in the mid to late 80s, who realized that certain diseases that has affected certain populations within the basal ganglia either led to a hyperkinetic state, moving more, or a hypokinetic state, moving less. And chief among these would be Parkinson's disease. And so that bedrock serves as the bedrock for most of the existing models right now, where myself, and this is not anything that I invented, I'd say the truest to that model, and one that's been championed by John Krakauer, David Robbie, Rob Turner, who doesn't get nearly enough credit for the brilliant work he does, is that the basal ganglia serves as a modulator of vigor. You can think of vigor as force over time is one way to think of it. There are other interpretations thereof, but modulating the kinematics of an action that is selected upstream, presumably with motor cortex and its friends. And that's made sense to us in probably the biggest paper we've had so far. I stimulated either the direct or indirect pathway of the striatum. And these are two populations of opponent neurons in the striatum of mammals as well as most vertebrates. And the idea there, that what we found is that we could change the speed of a mouse reaching using one of these joysticks. That if we stimulated on fast reaches, this direct pathway population, we would get more fast reaches. We wouldn't necessarily speed up that reach, but we'd bias the animal in its performance. And if we stimulate on slow reaches, we got more slow reaches. The opposite was true for this indirect pathway population. The chief alternate hypothesis, which is most often taught in textbooks or sometimes just put right next to it as if it's the same thing, is that when it comes to the motor side of things again, that the direct and indirect pathways are pro-movement and anti-movement. And this is where the gating idea that you introduced comes about, that you would that cortex and its friends would have several different possible actions that could be performed at any given time. One researcher theorized it to be around 4,000, and that through these parallel action channels, the striatum selects, the direct pathway selects which one behavior to do, and the indirect pathway selects the 3,999 to suppress.
Where did this number come from? Do we know?
It was based on a computational model of just what's possible. Okay. So not necessarily that the brain necessarily is doing it, but you suppress a whole lot of things that you don't want, and you facilitate and promote the action that you do. And some of the leading evidence. So let me back up. This was very attractive from a computational standpoint, and much of the support for this comes in the form of computational models, where the connectivity inside of architecture lends itself well to an action selection approach. But if the evidence for it, which there's plenty of it, probably the best is Lex Kravitz, who's at WashU, work that he did while he was at UCSF. He stimulated the direct or indirect pathway just as I did, but left the laser on for 10, 20, 30 seconds. And in this case, those neurons that were optogenically stimulated, if it was direct pathway stimulation, the mouse would start walking after a few seconds, and start and walk. And if you stimulate the indirect pathway, the sort of stop pathway, presumably because you were gating off all action, the animal would stop locomoting and wouldn't start again. This is fine, except not that any optogenetic stimulation is particularly physiological, but
Sure.
These neurons are only active very infrequently. Most of them have baseline firing rates, at least in a mouse, of under 1 hertz. And they fire very specifically around movement onset, at least in trained behaviors, as well as locomotion. And so that's just sort of odd that you need to stimulate for these longer periods of time. Also, the fact that it was limited to locomotion. If you stimulate one side, it's as if you're applying either the gas or the brake to the contralateral side of the body, but not the contralateral side of the body. It's the turn. But in order to turn left or right, you need both your left and your right limbs and your head and all the postural muscles of the torso. So that just sort of struck me as odd. And so that was a lot of the impetus for the very brief and very selective stimulation that we did. So if we stimulated on every reach or on a subset of reaches, we observed no effect with these under half-second pulsed light to sort of just try and nudge the system at a few more spikes when the neuron would normally be active. We found no effect on that. And since then, we've managed to expand this to starting and stopping. So in work that we should be submitting soon, we looked and saw if we stimulate the direct pathway every time a mouse stops locomoting in the open field, the suggestion of the gating pro and anti-kinetic action selection side would be that you should, just as Lex demonstrated quite well, if you stimulate that mouse should stimulate direct pathway, that mouse should take off walking,
Start moving again.
Yeah. What we found was demonstrably the opposite. A mouse will stay pretty much seated for minutes at a time if we stimulate every time the animal stops moving. So looks a lot more like reinforcement and along a kinetic axis of in this case locomotor speed. An indirect pathway has the opposite effect. The animal just more or less won't stop walking for the 20 minutes in which stimulation can occur. And I think what's really critical, and that I think is an informative point, is the causality is the wrong term, but while we stimulate, sometimes even though the direct pathway stimulation biased the animal to be stopped walking, to not be walking, sometimes we get up and walk in the middle of it. So we're biasing the performance. We're not selecting the action. I think
But you're stimulating. You're trying to nudge. You're not doing electroshock therapy.
We're not, at least as far as we can tell. So that might be part of it, but I think the interpretation that I think may be more appropriate with our causal manipulations and in what we see with our correlative measures, like recording, is that these populations of striatal neurons acts more as a gain modulator. So whatever signal, and this also has great benefits with the other motor or non-motor roles ascribed to the area. If you're just a gain modulator, and actually interesting you brought up cerebellum. Cerebellum and its wonderful repeated architecture serves as a computational unit that in theory can be applied to any input that it receives, and it's going to do its LTD learning wonderfulness on whatever the input is.
If there's any part of the brain that you could almost consider in isolation, the cerebellum would be it, right? Is it granule cells? Is that what?
Yes, granule cells. That's the little one.
Yeah, yeah. Whereas the rest of the brain is like so intertwined, and there's so much different crosstalk and recursiveness that it's just messier. But the cerebellum is clean in that respect, ish.
Yeah, and so our idea with this other subcortical, not that it is any cleaner in the connectivity sense, is that if it serves as a gain modulator, and if you know some basics of optimal control theory, if you have a push and a pull on any feature that you might want to control, it gives you a lot better control to have that push and pull than just the push. Some people say, oh, but I can drive my car with just the gas. If you needed to drive at 34.3 miles per hour, you would want to have a foot on both the gas and the brake to control that. And so if these neurons are acting as a gain modulator of whatever the incoming signal is, that applied in a certain fashion, getting back to how you ask your questions, that can look like action selection because of turning that volume knob, as it were, doing that gain modulation depending on the inputs, and that's viewed through a very specific lens. When looking at vigor, it's more obvious there that it would be doing that. And then plenty of people look at the basal ganglia and the striatum in terms of its cognitive or auditory processing or all sorts of things. And to get back at sort of these, what is the brain doing, and are there any sort of fundamental properties of neural circuits? It seems like you could copy and paste this computational unit of we've called it a history dependent gain modulator. So it's getting feedback information from dopaminergic cells and all sorts of convergent inputs from all over the brain. It would be a really good system. And our experimental evidence suggests it's a great way to pair descending inputs, feedback from your dopamine system, and to alter, to tweak those descending inputs, and either have it be a feedback system or in many organisms, a majority of the output of the basal ganglia is feed forward down to brainstem nuclei, superior colliculus, etc. For mammals. And so those same nuclei are getting convergent inputs from these cortical areas as well. So there's a beautiful paper that Josh Dubman wrote with John Krakauer. We also had one with the word history dependent gain in the title, sort of pushing this idea But yeah, that's where we stand. I still have a job, so it's certainly not figured out one way or another, and we're going after these things. But yeah, the general idea is that motor cortex, not necessarily M1 specifically, but motor cortex is saying what to do, and striatum is saying how to do it. And it actually, I think about my monkey work quite a bit, because even in a visual sensory motor area like LIP or parietal reach region, I knew 150 milliseconds or 300 milliseconds ahead of time if that monkey was going to saccade left or right, use its left or right arm to reach. And so just the timelines of the action selection model have never quite made sense to me. But
a gain, you can have a gain modulator that acts as a action selector in the extremes, right? So in a sense, I mean, this goes back to like, what does brain area X do? Well, it's not like one function, because it can, under those two alternative hypotheses, it can actually be both.
Absolutely. There's some reasons why, and I said, view through a specific light, it can look like action selection. I think there's some other more nuanced work that suggests that's actual action selection might not be what's going on. But
actual action selection as opposed to gain modulation action selection.
Yeah.
Okay. Yeah. All right,
action, that's fine. Something like that. It could certainly look like it, but if it's performing a selection between many possible actions, that
right. The way it's written up in the textbooks, that's the actual action selection you're talking about.
That's the better way to get at that, I think. Maybe we go back and edit that one too. But yeah, so it can look like action selection, but I think for selecting between dozens of possible actions, the evidence just isn't there, and we have a few lines of evidence that suggests that but that's also not the case. But it's still an open question. In fact, writing a perspective piece right now with several a dozen authors on just sort of trying to come to terms with these competing hypotheses. And some of the original, I talked about the bedrock theory on hypo and hyperkinetic modalities. Some of the people who came up with those models have said, guys, you've been using the same model since the mid 80s. Maybe you should get creative and move on to something else. But if you read a basal ganglia or a corticostriatal paper, you're almost required to cite Albin and DeLong. Brilliant guys. But yeah, there is some doctrine in there to deal with.
Part of what you've been talking about, especially with basal ganglia, and this holds for motor cortex as well, but your experiments on basal ganglia also showed this. And basal ganglia is also wrapped up with the story of learning, right? Very involved in learning. And part of your stimulation experiments showed that gain modulation is learned and lasts for some time.
Yes.
Either whichever way you stimulate, for the faster or for the slower, it lasts for some time.
It followed the policy, right? Which is policy learning is a beautiful form of learning.
Yeah. So I'm not sure if right now is the right time to pivot to. So this is all in the context of training animals to perform tasks, to move these joysticks, right? And then you do some causal manipulations, some stimulation, and you're recording neurons, and the organisms have to learn the task. They have to put effort into the task, etc. And so that's a large part of your history and current research is studying these tasks and how learning is affected and the kinematics and what's happening at the neural level. And you've taken kind of a recent turn, as has a lot of the field, into quote unquote naturalistic, was it neuroecological? Is that the term?
Ethological.
Oh, I say etho. Is it etho?
You said echo.
Did I say ecological? Oh. Okay.
We have not gone out to. We got invited to study elephants in a nature reserve, but we have not.
Right.
We've not taken them off on that.
Yeah.
Yes,
okay.
that one.
Yeah. Neuroethology. You say neuroethological or neuroethological?
For neuro, it's neuro to me, which is the last person you should trust.
Okay.
Especially as your employer. I say neuroethology, but it's neuroethological.
Okay. Anyway, the turn toward naturalistic behaviors, neuroethology. Yeah. Well, I wanted to say neuroethological behaviors again, but neuroethology encompasses, it's redundant to say behaviors again. Neuroethology behaviors in there, right? Where there is, so in the case of what you've done thus far, it's in your case, it's, letting a mouse just kind of explore a space, there's no task, right? There's no reward to earn. There's no skill to learn. So these are kind of like innate kinds of behaviors. And just recording neurons, right? In motor cortex and in basal ganglia and seeing how these areas and the axis of what you call the axis of M1 basal ganglia or motor basal ganglia axis, seeing what's happening in these brain areas during these naturalistic behaviors. And so I don't think I've ever asked you what you expected to find in the first place.
So I don't know that I had a real expectation of what I wanted to find. The motivation behind it really is what we were just talking about, of if this is if the basal ganglia do action selection, or if cortex says what action to do, the fact that our behavioral space for actions is to press a lever or not, or to move a joystick, to turn left and right, which is sort of baked into the problem of if you stimulate for 20 seconds and the only behavioral output in that paradigm is it walks more. It takes a little bit, but it walks more.
That's something. Not to denigrate that. That's something.
It's huge.
Yeah.
And at the time, that was 12 years ago, that was monumental. It still is. But part of the motivation to get into more ethological behaviors and more, I'd say our focus is more on the spontaneous side, the untrained part of it, is because now the action space that you're selecting from is one, much bigger. It's not turn left or turn right. It also is unencumbered by the potential caveats of training or overtraining, or the fact that most mice don't see a joystick as they're walking through the woods. So that was a big motivation to try and figure out what these circuits are doing, is to let nature make a lot more of the decisions. So that opens up our behavioral parameter space, which may open up our neural dimensionality. And we have some initial evidence that is the case. Not as big as I thought, but that sort of remains to be nailed down.
I'll interject here and just say, just as a point of, I guess, education, and we talked about a lot about it on the podcast, but modern neuroscience is kind of enamored with these dimensionality reduction techniques that work especially well in constrained laboratory tasks like reaching, where there is a very specific movement that an animal will make in various directions, right? And then you take a high dimensional data recording, let's say thousands of neurons, and you can use dimensionality reduction techniques. And then instead of reading out the 1,000-dimensional neural activity, you reduce it down to a few important dimensions based on the variance of how all these neurons are covariating with each other, and et cetera. And then you can plot out the trajectory of this low-dimensional trajectory. And there are distinct trajectories for, let's say, different arm reach directions, right? And this has been kind of a powerful way to consider, especially what's happening in motor cortex and in motor areas in general. And why did I interject that? I interjected that because, So I highlighted that that's been very effective, especially in these constrained tasks. And so it's an open question of whether in these unconstrained kind of naturalistic tasks that you're going for now, whether that same kind of dimensionality reduction approach will be as effective, or what will come of it, essentially.
Right. And we know that when it comes to just simple, in the most basic and maybe abstract form of neural encoding, we know lots of neurons have firing rates and responses like lots of other neurons. And this redundancy of representation almost has to happen, no matter how you think the brain is organizing itself. We are losing and gaining tons of neurons all the time. As adults, it's more on the losing side. But we aren't fundamentally different because of that. And so the idea of saying, hey, can we take these 1,000 neurons that are, some of them are more like each other than others. They're all their own, maybe special snowflakes, but we can get a composite, distilled down summary out of these neurons. I think has to be the case no matter what. But you're right. I think when it comes to the dimensionality of representations, as well as the complexity of these dimensions, people like to talk about manifolds, which is sort of the shape that these neural trajectories take on. We don't know about the complexity of how many dimensions, how many of these, if you think of each dimension as a feature that may be repeated or shared to some extent between several neurons. We don't know how many there are and how complex that is sort of laid out. And that's one of the reasons that we're getting into this, because if you train an animal to do a task and they only have one thing to worry about, it's reasonable from a strictly hand-waving perspective to think, well, we might not be getting the whole picture. The brain is sort of in this, I don't want to say default state, that term's taken, in the simplistic state of just needing to do whatever you're going to do. And we only hear from active neurons anyway, so there may be plenty of other neurons that are representing other dimensions of behavior or whatever else is going on for the brain that you'll just never see in this format. I'd say one other part is that that overtraining itself, besides the fact that these are typically are very simple things, the overtraining part of it, the practice and practice and practice, may have and almost always goal directed, I think is a factor that is rarely brought up. It may have profound changes on how we interpret those signals. And it's been known for years that if you put your electrode in the brain early on in training, and this is across species, across many tasks, and even if you try and match the kinematics of individual trials as best you can, early on training, you do not get the pretty response profiles that you associate with well-trained tasks at a single neuron or population level. Are they there? Maybe. Something has to be going on. But it's not very obvious what's going on.
It's not like you were, let's say it's a reaching task. It's not like you were unable to reach before. It's just that you haven't been reaching in that context, under that goal directedness, in that specific location that you need to be within a few millimeters of a certain area to get a reward. And so there's a lot going on. You're really shaping the brain in that case to reach in a certain manner. So it's like you're creating something to study it. So you're not studying reaching or reaching behavior in general. You're studying reaching behavior in a very specified goal-oriented task under reduced conditions, et cetera. Right.
Yeah. And I
don't mean to pick on reaching in particular.
Yeah. I mean, that task is still being done in our lab, and there's some great work, but just to make my perspective clear, you said that a little bit sort of tongue in cheek that this is way too abstracted and there's all these potential problems. Once again, the right approach for the right question is fantastic. And so we have a senior grad student, Mark Nicholas, who is getting after this. What does motor cortex do, and what does striatum do, and lesion motor cortex in a reaching task? And lo and behold, without M1, if you look immediately after and in the days after that lesion happens, mouse can't do that task. It can still walk. And so in the second part of the paper, we actually look at a mouse walking down a hallway. It walks pretty well until it has to make a decision. And Sylvia Arbor and several other groups have similar effects to this. But in this case, again, a trained artificial task can be very useful. In this case, we're trying to complement both sides of it, of the well-trained and the spontaneous. When we have the animal walking down the hall, there's no reward. But I will say, you listed off, the different goals of you have to be within a centimeter of the target or the reward. All these facets that you just named a minute ago, and several more, are really important to us, and why we are going after these more naturalistic, spontaneous behaviors, and applying algorithms to try and give the same structure that we would have with our trained trials. And we've had a fair amount of success with that. But looking at, if we pull that away, And we'll soon be starting to sort of pepper in some of those caveats that you mentioned of, oh, but what if this is a for reward? Or what if this is a fairly repeated within a certain context behavior? Trying to build a bridge. But you're right. In a sense, although I don't think things will be night and day different, there's a lot of opportunity for learning what the brain does in its spontaneously generating behavior state. And again, you were focusing on learning before. We're constantly learning. Sometimes we're focused on learning or not. But if we can't learn a particular behavior, learn how to interact with something, our fitness goes down, and we're not going to pass on our genes. So this is the tasks certainly have their place in neuroscience and in ethology itself, because it's not that
sure, yeah,
this animal is created as an adult with everything it's ever going to do, and it's never going to update according to the chaos and conflict in its environment. Yeah, so it's all important.
One of the things that you said to me maybe months ago, I don't remember if you said this the first time we met.
You haven't been in the lab that long.
Well, it's been a few months, but anyway, it has stuck with me, is that it's a very simple point, but it really stuck with me. We're like, you and I right now are behaving. When you're sitting down doing nothing, you're behaving.
I'm doing so many things when I'm sitting down doing nothing.
You are. Not everybody is. You are. We know the octopus is doing nothing when it's sitting, but yeah. No, but I mean, I think that's one of the challenges, right? Is, for example, what is a behavior? Which is a very simple question that we don't have a great answer to, but it's been, studied philosophically for a long time, et cetera. But, just the fact that you and I right now are behaving, and presumably, our brains are have something to do with that behavior. Let's say that there's a correlation between our brain activity and our behaviors right now, at least.
Let's hope so.
Yeah, let's hope so. And so when you have a mouse just, it's kind of wandering around and exploring, naturalistically behaving, a lot of the times just kind of sitting there and might kind of turn a little bit. And there are all these nuances that are that neuroscience for decades has sought to get rid of, so that we can control exactly what we can measure. And so that we're measuring a repeated behavior over and over, so then we can average the trials together and look at the average neural response, and then compare conditions. And if these 30 trials in this condition, then we can compare it to 30 trials in that condition. But then all of a sudden you have to kind of throw that out the window, which I think comes with challenges, and is exciting as well. Yeah.
Yeah. And so you hit on a couple really important points there. We are constantly in the middle of doing a behavior, and it can, even the behaviors that we think we understand, it can be very complex. To get back to reaching again, in order to extend your hand out or to pull it back towards you, we always think, oh, there's shoulder and bicep, and you're engaging all of your trunk muscles and the opposite
core.
side
That's
of the
why
body.
my core is so strong. I'm reaching a lot. That's how I work out my core.
Your core is probably far better than mine. And I'd love, I teach systems neuroscience to the undergrads here. And I love the example in the book that if you're to pull a door open, the first muscles to engage are your calf muscles. Because they've got to be able to pull you backwards so that you can pull against the door.
Otherwise, you really just got to pull, you're going to slam into the door.
I've done that too. More often it's the pushing and the door fails to move because I don't know how to read. There's a Gary Lawson,
one of my favorites.
cartoon. Oh yeah. School for the Gifted.
School of the Gifted. Yeah. Anyway, to hash that, to flesh that out, the door says pull, I think, and he's pushing on the door, and then above the door, there's a sign that says School for the Gifted, something like that.
Common sense ain't so common.
Yeah.
So right, we're constantly in a behavior, which does make some of these tasks a little bit curious. That we think of the behavior as starting at point X, and then once they get to the target, it's done. Same thing with learning. Many people in the lab have probably heard dozens, if not hundreds of times, that the fact that we're always updating our internal models, we're always learning, and that I get why some papers will say, oh, learning is here and performance is here. There's going to be different sort of times when there's more focus, but it's more likely how accurate or how much do you have to update your model. I think you're constantly tweaking and updating, and there's good evidence for this. So yeah, there's a lot going on, and in terms of all of those nuances, we do our best. So a few years ago, an insanely gifted graduate student, Alex Su, we came out with a platform called B-SOiD, spelled B-SOiD, which all sounds the same if you say it with a Brooklyn accent. I'm not going to do it on this show.
Oh,
I know better than that. But you can do it if you'd like to.
I'm all right, thanks. I really wanted to hear you do it. I would butcher it, so
I did just a hair of it.
Brooklyn. Okay, yeah.
I think. Anyways, that takes pose estimation data, for instance, deep cut, sleep, deep lab cut, open pose.
These are modern tools that yeah. Well, you'll do it better than I do.
Computer vision algorithms that reduce the thousands of pixels into a video down to key points that are essentially motion capture suits on an individual, but does so with modern computer vision techniques. And so we can take those data, which are very informative, much more informative than the average pixel in a video. We look at the spatial temporal pattern of those movements, and anyone who has seen a motion capture suit, you can tell when someone is walking or jumping up and down, just by the 12 points that you see on the body. And so B-SOiD is an unsupervised learning algorithm that identifies spatial temporal patterns in those points and adds interpretability to the pose estimation data by extracting those spatial temporal patterns that we would call behavior. The code itself will work on anything where you need pattern recognition. So we've used that to get at trying to identify behaviors. And
what it ends up spitting out is essentially categorical variables that correspond to these kind of repeated motifs that you might consider locomotion or turning left, turning right, things like that it groups, that it clusters into these common motifs, right?
It only knows it as group 2, group 12, but it turns out quite nicely that they tend to map on quite well to established ethological behaviors, like locomotion. We actually there's sort of 3 canonical types of grooming a mouse will do, of the sort of the face, the bigger motion of the head. If you think of a cat or a dog sort of pulling its ear down to clean, that's the head groom, and then turning back to groom the body. We didn't tell it anything, and that's sort of the beauty of an unsupervised approach. Whatever you pull out has some measure of robustness to it, or believability in a sense. And we readily get those three different groups of behaviors. So we have that in our toolkit. We actually have a paper that just came out in Nature Methods for a supervised approach. So we call it A-SOiD. We're staying with the record dad joke of the A-SOiD and B-SOiD, where you might find a deep cut.
Man, that's a reach. I like it. It's a reach.
That's the basics behind it. Our emblem actually for A-SOiD is a record with an A on it. I was at a workshop recently, and someone said we should get, what was it? Get a version that's specific for fish and call it C-SOiD. Or we've got, that one I was both very proud and a little bit disappointed that I didn't come up with it myself.
Right. Yeah. I'm actually thinking of D now, but
D-SOiD might actually happen. So currently A-SOiD is both this unsupervised B-SOiD as well as A-SOiD, and you can D-SOiD, where you get into the behavioral segmentation side of things. But for more nuanced or specific behaviors, that supervised side can be really important. And it's both of them are powerful tools. Again, pick the right approach for the right question. But we've been using that to try to get us some sort of handle on these naturalistic spontaneous behaviors. And the fact that it maps onto what ethologists have been sort of honed in on for decades makes me feel better about the results. And that serves as a jumping off point for then, if we subdivide into longer or shorter grooming or rearing or locomotive bouts, or faster or slower, or other dimensions that we can make things more similar, we can get into those nuances, but we have a starting point. And something that in any talk I give on this, that I make a big point out of is, you mentioned before, we don't know what motor cortex is doing. I think we have a better idea now than we ever have, but especially for these naturalistic behaviors, we didn't have to have any sort of specific representation. And in fact, what we found is throughout motor cortex, dorsal striatum, and to some extent, even in ventral striatum, although the reasons why we think this, why we see it in ventral striatum, I think, are different. We have a robust response that's specific to each type of behavior. And certainly in our deeper layers of motor cortex and dorsal striatum, with just a very simple decoder, we can predict which of 16 behaviors a mouse is doing from just spike counts at a rate of around 60% with a F1 adjusted score, which is 1/16 is a lot smaller than 60%. So there's something there. It's not 100%. If it was 100%, I would know we were wrong. In part because that's just not how biology works. But
what if it's 103%?
Then that's not how math works. But yeah, it gives us some new abilities to ask questions about what is action, what is behavior, what in a naturalistic state that evolution is more often push these animals towards. How are these things represented? How are the decisions made? How does learning and adapting of these dimensions of representation change? And certainly, thanks to tools like neuropixels and other recording modalities, we can start asking not just what does a handful of neurons within one area, but how do multiple areas interact? And so we have complex, or I'd say a rich account of behavior and a rich account of neuroactivity, to try and get that abstract thought to action. How is that transformation made?
I wish you were busy this afternoon. I wish we had taken a few minutes. I could have shown you these trajectories in GPFA. It'll be tomorrow. Tomorrow we'll do it.
And great.
So
you'll just have to publish it.
Sure, you bet.
Real soon and discuss it on here. And yeah,
I'll come on the Brain Inspired podcast and talk about it. It struck me
nepotism at its best.
Yes, narcissism at its best. That would be.
Oh yeah, I guess for you,
I would interview myself, right? Yeah.
And I will say, if anyone's watching the video, I'm going to undo this. Paul specifically requested that
I did, and it was a half joke, but I'm okay. So you actually wear that when you teach. I put on a bow tie for teaching. I've loved bow ties for years, and it's a nice opportunity because I'll admit, one thing I love about academia, at least more on the science side than the engineering side, I can come to work in sweatpants, and that's what I normally do. Yeah, that's right. Yeah,
which bow ties and sweatpants. Although I do have a somewhat eclectic fashion style. I
can vouch for that.
I know better than bow tie and sweatpants.
Yeah.
I've learned over the years.
Well, thank you for wearing it. I appreciated that you didn't. I did it in half jest, and you actually came through for me, so I appreciate it.
You got to support you got to support the people in your lab.
That's right. That's right. So while you were talking about, some of the challenges and some of the tools that we have and how B-SOiD can categorize these things, and it turns out that it matches very well. The categories that B-SOiD comes up with matches very well to what has been described in the past by human eyes, by ethologists. And I was reflecting on our earlier conversation about artificial networks and using them to study things in cognitive neuroscience. And this is a case where behavior is a continuous, ongoing thing. And we're just talking about the syllables and motifs, right? And so in a sense, what we're doing is saying, well, how can we apply the old way of doing things to this new kind of continuous flowing behavior, right? So we want to break it down into things that we can discrete chunks that we can study, right? And say,
handles,
when does it handles? Yeah. When does it start? When does it stop? And in a sense, then you could use so what I was thinking is that an artificial network, well, you just turn it off and come back tomorrow. And you can't do that with behavior, right? Which is just a continuous, you can study it tomorrow.
Comas exist, but
you're not behaving during a coma? What about sleep?
Sleep is a funny one.
Anyway, before we get on, move on. Yeah. So I do want to come back to that. But it just struck me that,
have you had Emery Brown on the podcast?
I have not, but he's on the list. Yeah.
Yeah. Ask him that question.
Okay. Whether sleep is a behavior, is behaving. Yeah. Okay.
And the multiple consciousnesses thereof. Yeah.
Right. Yeah. His specialty. And coma.
Mm-hmm. I will say B-SOiD can pull out
coma.
We weren't we don't, we are not approved for an IACUC for that. It does pull out different poses. And we've noticed that there's different frequency band during sleep. We have noticed different frequency bands related to those poses. And I believe there's been a paper or two that through other means, I forgot if it was an automated method or not, has some correlation there. So Yeah, not the answer you're looking for is of behavior, but B-SOiD categories may apply to different stages of sleep. But anyways, you were going to ask something far
No.
more
I was just
intelligent.
noting that you can turn computers off, essentially, right? So using an artificial network, which you run a pass through, and yes, you can study the like dynamics, which has been when you study recurrent neural networks to compare them to the dynamics of like decision making in brains. You can compare the dynamics. But anyway, I was reflecting on the fact that artificial networks are static entities and living organisms are ever changing, ever behaving, constantly continuous behavior, neural activity flow. And it's just a fundamental difference.
Though the turning it off, I don't see it as that much of a difference. I'd say it's more the initialization, because also certainly an ANN or a DeepNet can continue to learn or run and change and do whatever it's going to do, given the opportunity and provide new information to it. But
I think I was thinking mostly in terms of it's going to spit out a discrete thing, and then it's going to spit out another discrete thing. And that's kind of what we're getting at by. So there's a tension in me about applying that kind of approach to studying something that I see as continuous.
And that is a big open question that I think will be one of the goals of this naturalistic behavior field moving forward, is how continuous is behavior, both behavior itself as well as its neural representation. Because although we can identify motifs for these individual naturalistic behaviors pretty well, surprisingly well. So I think we've made a decent foothold there. It's gonna be messy. It almost has to be messy. And I think then better word for messy, I think, would be complexity. And it may be that in some areas, spinal cord, there's not going to be a lot of messiness. There'll still be plenty of complexity. And there's constantly just incredible work being done on that side of things.
Good cover.
But it's
good recovery.
Oh yeah. I know what to do. But that's going to be a lot closer to the muscle, and it kind of has to be a more straightforward something closer to a linear operator. Whereas you get into motor cortex or these cognitive areas, where I'm sure if we plunk down an electrode into anterior cingulate or PFC, OFC, prior cortex, name your favorite area, I bet you we can still find a decoder for behavior. And who knows?
Sure, yeah,
V1.
sure. Yeah.
I wouldn't be entirely surprised if we can do well above chance from V1. It's not the first experiment I'm going to do. But in just getting at the complexity and the sort of fidelity to the discontinuous idea of you're doing behavior A and then you're doing behavior B, we can get a sense of how these are orchestrated and how that information is handed off from one area to another, and the computational processes that are going on. We have in this paper that's in the works, not only do we show that we can get a robust response from individual neurons for some amount of neurons for all these areas. Motor cortex is much more promiscuous in what it shows a tuning for in terms of the behaviors. So the average neuron in motor cortex, particularly layer 5, will respond to 4, 5, 6, even 8 of the 16 behaviors that we're keying in on. Whereas when we look at dorsal striatum, we see much more sparsity in its representation, talking 2 or 3 behaviors. And so these marked differences, which are sort of related to this idea of how continuous or discontinuous behavior truly is, I think are important, because at the end of the day, we got to do whatever we're going to do. But there's a whole lot of fudge room in there for far more complex things. We are not automatons. And so this is also part of my difficulty with the action selection idea, of the idea that discrete action channels, although that model has plenty of attractive things in terms of a theoretical computation that's fun to play with the math, or as well as a useful framework through which to view brain and behavior, how you would actually form an action channel and the discreteness that implies. I'm not saying that my half-baked ideas are any better, but though that has always rubbed me the wrong way.
Fair enough.
So yeah, we'll see just how discontinuous behavior is, and almost certainly different behaviors will occupy different parts of this complexity spectrum. And so, yeah, we're actually recently struck up a collaboration with Jose Mora, who's one of the fathers of recent signal processing work, and I'm excited to have that. And really, anyone who is capable of asking some of these questions that I think are, like any question, in neuroscience, there's going to be some assumptions, but there's a lot of openness and unknowns that I think we can make some real headway on. So that's, I imagine, more and more in our lab we'll focus on this naturalistic side, but it's still asking the question of how these circuits locally and in aggregate lead to the behavior of interest. It's going to be exciting for a while.
Oh, okay.
And not just us, so many cool people doing it.
Yeah, there's a lot of people doing it. That's for sure. It's going to be exciting for a while.
And then I'll move to octopus.
And then you'll move to octopus. We're in kind of I probably every scientist says this in the midst of their quote unquote time, but it seems like we're in an exciting time because computationally we're way more powerful, all of us, in the past 10 years, and it's just continuing to increase. We can record more neurons than ever. We have more tools, optogenetics, right? We can track neurons and stimulate them and watch them. So on the other hand, it opens like challenges of how to use the tools correctly, how to ask the right questions with the tools, right? And it's like a confluence of all these things. And now the move into naturalistic behavior. So there's a lot to look at, right? Or neuroethological behavior. There's a lot to look at with these new tools. I did that without a hitch. And earlier I asked you if there's anything you missed about the old days, right? Working with non-human primates, with recording less neurons, you'd go down with a single electrode usually and just listen for a neuron that sounds like it's doing what you wanted it to be doing, and you record it. And there's something wonderfully simple about that. And things are not as simple anymore. Everything's a low dimensional manifold from, recording your high dimensional data, et cetera, et cetera. And there's a lot more statistical analyses available. Does a part of you wish that you were a neuroscientist 100 years ago? Or, would you, if you had to choose, would you go back and be a neuroscientist 50 years ago? Or would you be right now? Or would you be 50 years from now if you had to transport yourself, or not?
As a curious person, 100 years from now is ideal because we'll know a whole lot more. I don't care if I'm right or wrong.
I love that about you, by the way.
I think it's a critical part about science, but as it was phrased to me by one person, someone disproved it. It was during grad school, Greg DeAngelis, while he was still at WashU. It says someone cared enough to prove you wrong, and is still reading your papers. But no, I want to know what the answers are as a curious person. So yeah, I'd pick the future because it'll take a while. And I think you mentioned our experimental tools being very robust. We have Carson and others
Carson Stringer.
Recording tens of thousands of neurons with calcium imaging, and Allen Institute and all the Josh Siegel and dozens of others with all their electrodes, Nick Steinmetz. So we have tons of, or even just with the single neuropixels probe, we have tons and tons of neurons and constantly more complex behaviors, trained and otherwise. The annoying question that I like to ask people is, if you could record action potentials from every neuron in the brain, how would that change your science?
Classic annoying question.
Oh yeah. And typically the best answer I get is I wouldn't have to do as many experiments because I would have my N for neurons would be very, very big.
Set. You'd have the only data set you needed. Yeah.
Which is great. But yeah, I will say that I hope and I'm optimistic that in the next few years we will have more tools to deal with as the volumes of data that you pointed out, which are made possible by new experimental tools. But I think to the answer that I was about to say is, as a neuroscientist, yeah, I would prefer 50 years ago to 100 years from now.
Oh, that's interesting.
In part because of what I find really exciting. I mean, if you look 50 years ago, would have been, oh, that's a scary thought. 1974.
I know it's already like late in the game in terms of a lot of things that are like the classic things that have already been discovered, essentially.
Yeah. So I mean, I still think 2005 was 6 years ago, but that's a different topic.
Yes.
But
yeah,
think about all the titans, Mountcastle, Everett, Georgopoulos, Wirtz. These are mainly motor people. Eccles, speaking of Cerebellum, although most of his stuff was a bit earlier. You've got all the great stuff in V1 still sort of happening, though I guess still a little bit earlier. Yeah, you have all these pillars of findings, and sort of in a very small way. And also, to be clear, I am not saying that I am on par with Eccles, but the sort of questions that all of them were going after are what really would excite me. I mean, I just showed video of Hubel and Weasel with their light beam. That's sort of the classic. They move it across the screen, and you can hear each action potential from a neuron in V1. I got so many weird looks setting up my lab. I said that I need every amplifier to come with a speaker so I can listen in on a neuron if I want to.
Why is that weird? You're speaking to the choir. You're preaching to the choir.
Yeah, it's a holdover from platinum iridium single unit electrodes in a monkey. But it is so satisfying. I'm glad to have my multi-channel electrodes. But
that's true. No one hears very many.
What would you listen
people,
to?
Yeah, hear the
You'd
popping.
have to tune it into just one of 384 channels.
Yeah,
which you can do.
You can.
We've done it in lab. Yeah. But the sort of questions that they were after and the way they went about it, there is something. I don't think it's a romanticizing it, but I think there's the questions and the approaches that I think are a whole lot of fun are when it's kind of like what we have for naturalistic behavior. There's as you said, it's exploratory. We don't necessarily know up from down, and that there are likely, hopefully, knock on wood, some low-hanging principles that probably low-hanging fruit of principles that we may be able to get at that, years and years in the future, there's still the new principles to be found, but they're more niche. You've moved down the dendrograms of your subfield more and more. So there's something sort of exciting to me, the creative approaches. I mean, really the same single unit recording that we're talking about, it was still in the infancy 50 years ago, which is something that we don't necessarily, I mean, we sort of take it for granted, or saying, what is parietal cortex doing? We've had some of this in a clinical sense, and Brodmann areas, etc. But yeah, guys like Mountcastle just marching around and saying, "This is what I see." It's a very cursory.
I know, but there's something so nice about being able to do that. All right, Eric, this has been weird and fantastic.
We're not that weird.
No. You didn't make it weird. It's just I'm about to say two things to you that I've never said to a previous guest. One, I'll see you tomorrow. And two, I'll bring the octopus.
If it's to eat, don't. I'm allergic. Please don't.
All right, thanks for doing this.
I hear it's delicious. But yeah, you twisted my arm, but this was a lot of fun. And hopefully someone else thinks this is intelligible and maybe even interesting.
I alone produce Brain Inspired. If you value this podcast, consider supporting it through Patreon to access full versions of all the episodes and to join our Discord community. Or if you want to learn more about the intersection of neuroscience and AI, consider signing up for my online course, Neuro AI, The Quest to Explain Intelligence. Go to braininspired.co to learn more. To get in touch with me, email paul@braininspired.co. You're hearing music by The New Year. Find them at thenewyear.net. Thank you for your support. See you next time.